Postsynaptic potentials that are caused by neurotransmitter chemicals can be either depolarizing, often but not always resulting in an excitatory postsynaptic potential, or EPSP, or hyperpolarizing, resulting in an inhibitory postsynaptic potential, or IPSP. Postsynaptic potentials generally move passively along the dendritic membrane, gradually becoming smaller as they spread. Therefore, the postsynaptic potentials from more distant synapses will decay more than PSPs from synapses closer to the integration zone at the axon hillock. The postsynaptic potentials produced at most synapses are usually well below the threshold for generating postsynaptic action potentials. How then can synapses transmit information if their postsynaptic potentials are subthreshold? Suppose that two excitatory endings are activated, causing local depolarizations of the cell body. Taken alone, neither would be sufficient to trigger an action potential, but when combined, the two depolarizations sum to depolarize the membrane in the hillock region to threshold. When inhibitory synapses are active, the membrane potential tends to be stabilized below threshold because they induce hyperpolarizations, or subthreshold depolarizations, that cannot reach threshold. These postsynaptic effects also spread passively, dissipating as they travel. Because some potentials excite and others inhibit the hillock, these effects partially cancel out each other. Thus, the net effect is the difference between the two. The neuron subtracts the IPSPs from the EPSPs. Postsynaptic effects that are not absolutely simultaneous can also be summed, because the postsynaptic potentials last a few milliseconds before fading away. The closer they are in time, the greater is the overlap, and the more complete is the summation, which in this case is called temporal summation. The summation of potentials originating from different physical locations across the cell body is called spatial summation. Only if the overall sum of all the potentials, both EPSPs and IPSPs, is sufficient to depolarize the cell to threshold at the axon hillock, is an action potential triggered.